The overall goal of this procedure is to monitor the level of intracellular glucose or glutamine non-invasively using genetically encoded protein-based biosensors. This is accomplished by first transfecting the cells of interest with the biosensor gene-containing plasmid and selecting for those cells expressing the biosensor. In the second step, cell culture samples are taken from batch or fed batch cultures and their FRET ratios are measured. The intracellular glucose or glutamine concentration of the samples can then be determined by an independent assay. Ultimately, the FRET ratio of the samples can be used to calculate the intracellular concentration of glucose or glutamine non-invasively and in real time.
The implications of this technique extend toward the improvement of bioprocesses because it is a reliable method for obtaining real-time metabolic information that can be used for online optimization and control.
Though the method can be used to provide insight into bioprocessing, it can also be used in other systems, including the sensing of disease markers.
Begin by reviving CHO cells in 9 milliliters of complete growth medium. Then, spin down the cells, resuspend the pellet in 10 milliliters of fresh growth medium, and count the cells by trypan blue dye exclusion. Next, initiate a culture at a seeding density of 3 × 10 to the 5th cells per milliliter in 125 milliliter shaker flasks at 37 degrees Celsius and 5% CO2 on an orbital shaking platform with a 125 RPM rotation. Subculture the cells every 3 to 4 days in the complete growth medium at a seeding density of 2 × 10 to the 5th cells per milliliter.